Hello friends, in this video lecture I am going to talk about the physiological states of the F factor and what are the different types of factors are there and what are the meaning of those factors during this conjugation process. Okay, because those things are really important for learning purpose. Okay, so let me just take the tools. Okay, fine. Now here let me take a color. Okay, fine. Now let us talk about the definition of conjugation first. The conjugation means the gene transfer from one donor to a recipient by by direct physical contact between the cell. So here we will see in this picture that there is a direct contact between a bacterial cell. So two bacteria, it's a bacteria A which have to be a donor, and bacteria 2 which has to be a recipient for that gene, and then they will interact with each other by direct cellular contact by making a tunnel through them, which is made by their structural protein called pillins, and through this tunnel they can transfer their genetic materials. Okay, now there are different mating types of bacteria, so there is a different things called the mating types in between those bacteria. Now there are different types of mating types called F type or HFR strains, or there are different names you can heard of. Most of the time students create confusion in their mind about these terms, but I am going to clear these things out for you. Now here if you think about the donor cell, a donor cell must have something before the conjugation process, and that is called the F factor or fertility factor. Now what we mean by this fertility factor, that is denoted here. This is the fertility factor of this. Fertility factor means it must have some extra chromosomal part. Remember, extra chromosomal part, not the part of the chromosome. It's an extra chromosomal part. So it must have the extra chromosomal part with it, and this extra chromosomal part is often called plasmid. Now this plasmid must not contain the necessary ingredient for structural support of the bacterial cell, but must have some other advantageous genes. That means, for example, sometimes a drug resistance gene, sometimes the gene for uptaking some other type of glucose or uptaking some complex structure of glucose. Okay, so these things can be here inside this extra chromosomal gene. Now this is called the F factor or fertility factor. Now this factor have to be present there inside the donor cell before going through the conjugation step. Otherwise the conjugation cannot be established, and that's not at all but another important thing about this fertility factor is that fertility factor must have ORIT or origin of transfer. So from where this DNA material will be cleaved and can be transferred. So that is called the motility factor for that plasmid. Okay, because non-motile plasmids cannot participate in this conjugation procedures. Okay. Now if we think about the recipient cell, which is denoted here, the recipient cell lacks that F factor. So that is why the affinity between this donor and recipient happens, because donor is having this F factor, recipient is not having the F factor. So they will come and this recipient will take the F factor from donor, and by taking it, it finally receives that F factor, and it will become the F factor or fertility factor containing plasmid. Okay, now this F factor have another important thing to do, is that F factors can produce the sex pilus. What we can see here. So you can see in this case the donor only can form this pilus, which is a tunnel which is made by the pili proteins. Now this tunnel is only been made due to the presence of this F or fertility factor. Except for this fertility factor, we cannot produce this sex pilus, what you can see in case of recipient cells. Okay, now let us move on to the physiological states of F factor and different types of F factors. Now the first one, what we call the autonomous F factor or F plus factors. Now what we mean by this F plus factors, if we look at this picture, that the characteristics of this autonomous factor is that F plus, here we can see it is having this extra chromosomal DNA, which is carrying all the fertility factors or containing the origin of transfer, and also it is having the important regions which can be transferred from this donor cell into a recipient cell. Okay, now in this case there are low transfer of donor chromosomal DNA into the recipient chromosomal DNA. That means this chromosomal DNA is not being transferred. What is being transferred is this is this extra chromosomal DNA, or F factor, right, or fertility factor can be transferred. That's the important part. So that is why it is called the autonomous, because it do not depend on other things, so it can do its own job for this purpose. Now if we think about another factor which is called the HFR factor, it is also called the high frequency of recombination factor. Now this high frequency of recombination factor can be formed from this F factor. So you can see there is a F factor, so F plus means it is having an F factor. Now this is integrated. Now this plasmid DNA is integrated into the chromosomal DNA in somehow, or due to some crossing over event, due to some recombination event, this F factor is become conjugated with the cellular chromosome of the bacterial cell, and right after that it will form a large chromosome which is containing all the structural genes for the bacterial cell, all the chromosomal DNA of bacteria as well as the plasmid DNA of bacteria. So having both in the same cell, we call them the HFR factor, we call them the HFR type of bacterial cell. Now we can call it the high transfer of high recombination frequency, or high frequency for recombination. Why? Because if we think about the recombination of chromosomal genes, then in that case this frequency for this type of recombination is really high. Why? Because as they are having this blotches of this fertility factor, it is having the ODT, so it can be clipped from the ODT and it can be dragged onto the recipient cell. So that's why, due to the presence of this high frequency, due to the presence of this extra chromosomal DNA as the F factor, along with this chromosomal DNA of the bacterial cell, it can be easily transferred. Okay, so that's why the normal frequency of recombination of the chromosomal genes are high, because if we create a cut in this place and can drag a DNA strand from here, it will after some time it will take the chromosomal DNA with itself, right? So that's why, due to the presence of this extra chromosomal DNA along with the chromosomal DNA in this kind of cells, it actually increases the frequency of recombination for the chromosomal genes. Okay, now let us move on to the last and third type of a factor we will talking about, and that is called the autonomous with donor genes factor, or called F prime factor. Now why and how this F prime factor generates? If we think about the HFR strain, so this is the HFR strain, that means the chromosome with the blotches of extra chromosomal DNA. Now sometimes what happens, due to the normal recombination event, this kind of thing is formed, and sometimes there is a strand exchange between themselves, and also sometimes reverse recombination event to drag some amount of these things up. The actual goal for this is to make this plasmid DNA separated from the chromosomal DNA, because sometimes cell need to separate the plasmid DNA from chromosomal DNA, and during this process there will be fault. There is they make different mistakes. As a result of this mistakes, sometimes what they can do, they start to take few stretches of the chromosomal DNA with itself instead of the plasmid DNA, as you can see in this picture. So as it is trying to drag this extra chromosomal DNA along with itself, it drags some amount of chromosomal DNA with itself, and it leave some amount of extra chromosomal DNA with this bacterial chromosome, and as a result it will form a mixture type of extra chromosomal DNA which is having a small part of this chromosomal DNA. Okay, now carrying this small part of this chromosomal DNA again increases the chance of the transfer of this segment of this chromosomal DNA. So again in this case we call it autonomous, because it can be transferred on its own, because it produces the separate entity from the chromosomal DNA, but still due to the presence of this chromosomal DNA stretch onto itself, this is not totally autonomous, so we call it autonomous with donor genes. Okay, so we have seen three different species of bacterial cell, not actual species, three different types of bacterial cell. In this case you can see one is F prime, which is having in F prime cell, so take home message, in F prime cell we are having a bacterial cell. Inside the bacterial cell we have two things, one chromosomal DNA which blotches which small sequence of the extra chromosomal DNA, and one plasmid DNA with small stretch of the chromosomal DNA. Now in HFR strain, what we are having only one integrated chromosome and plasmid content. In this case, we are having the chromosomal DNA of bacteria, which is integrated along with the plasmid DNA of the bacteria. Now in the third case, what we are having, the in case of F plus, we are having both them together. So we are having one full chromosomal DNA and one full plasmid DNA. Okay.